So welcome back to the Immunology Classics of Immunology Journal Club. Before we get started, please like the video and click on the bell and check out the website and particularly the writing section of my website. So today, we're still trying to figure out how T cells help B cells so much. And it's a favorite topic of mine because of the fact that it implies that B cells don't know what to do with themselves. They need a little help from their friends. So we're on to the third factor that helps B cells, that is T cell derived. And the authors of this paper came out in Nature in 1986 in November the 6th, back to back with last week's paper, which was about Interleukin-5. This one is about Interleukin-6. And the authors are Toshio Hirano, and then 14 more authors. And then the last senior author is Tadamitsu Kishimoto. The title is Complementary DNA for a novel human interleukin that induces B lymphocytes to produce immunoglobulin. Now, the background on this paper is, I don't want to be too redundant, but I think it's important to fill it in from going way back. So the first, in 1970, 69, 70, there were a couple of papers out that show that when they tried to activate spleen cells in vitro with sheep red blood cells as antigen, that if they took out the T cells, they didn't work anymore. No, you didn't get any immunoglobulin. And so then that set the stage for it. Then in 1971, Dick Dutton, as a consequence of the International Congress of Immunology, he had a conjectural paper suggesting that perhaps there were soluble factors that might solve this problem. And then Anneliese Schimpel and Eberhard Wecker in 1972 published a paper in Nature New Biology that we reviewed that indicated that there might be something that was called a T cell replacing factor. As to what it was molecularly was the question. That set the stage from 1972 on, people trying to figure out what was happening. So this is now 1986. That's an awful lot of years. As you know, if you were an old geezer when you started this whole quest, you'd never see the end of it. Right after that, in 1973, it turns out that Tadamitsu Kishimoto was a young postdoc with Ishizaka. And as you recall, Ishizaka, the Ishizaka man and wife team published a landmark paper in 1967 that we also reviewed here, where they discovered that there was an immunoglobulin molecule isotype called IgE that was undiscovered, but that was the crack in the wall that uncovered what was going on with allergies. But this time, then in 1973. Kishimoto was a postdoc with Ishizaka, and they did a series of experiments at the Good Samaritan Hospital in Baltimore. Ishizaka set himself up, not at Johns Hopkins, which would have been the natural thing to do, but, there was a lot of sort of, I don't want to say bigotry, but there was a lot of racial kinds of things. Back in those days, the Americans didn't see too many Japanese people in science. So anyway, he had a nice laboratory there. And I knew all about that because I also happened to be in Baltimore between 1970 and 1972. I was a clinical associate with the National Cancer Institute. They had a branch there. So I would pass the Good Samaritan Hospital on the way down to work. But this is before I really discovered immunology. I sort of knew that these guys were there and they were doing this immunology work. Well, this turned on Kishimoto. Kishimoto became very excited about this whole process. And this is the beginning, really, of the involvement of a whole Japanese mafia of scientists that over the course of the next 20 years really were hot on this interleukin field. And so there were much fewer, even though there were a heck of a lot more immunologists in the United States of America. Most of them, they didn't want to know about that. They wanted to know about what the T cell antigen receptor was, and they wanted to know about what this so-called MHC restriction was. And lymphokines, there was lymphodirect to them. There was one other guy, which is Bill Paul, and I'll mention him in a minute, that was at the NIH and the Allergy and Infectious Disease Institute, that was into cytokines and lymphokines and that sort of thing. And he was into this B cell area as well. or he became that way in the early 80s. Now, Kishimoto and Ishizaka had shown that there was a soluble T cell factor that they could show that if you activated mouse spleen cells with a hapten carrier conjugate, carrier being a large protein molecule and a hapten being a little chemical appendage attached to the carrier would activate a lot of T cells. And though the T cells then would make a soluble factor that somehow their experiments showed that it would work on hapten primed B cells and make the B cells then make immunoglobulin. That's what Kishimoto got excited about. That was in 73, and 74, Takatsu, whose paper we reviewed last week, came out with a TRF kind of a molecule. And Kishimoto left, after he left Ishizaka's lab in Baltimore, he went back to Osaka. So then Takatsu, he was in the southern island, Kyushu. So they weren't all together back in the 70s. It was only later on where they got to be really hot and heavy on all this stuff. In 1982, then, 10 years later, after Schimpel and Wecker's paper, Bill Paul at the NIH worked up a system where he activated splenic B cells with anti-mu, an antibody, to the heavy chain of the IgM. And then he could add his T-cell derived factor, and he could then measure increased proliferative activity in the B cell target cells. At the same time, Ellen Vitetta down at the University of Texas at Dallas, Southwestern, showed that if she activated splenic B cells with the lipopolysaccharide and added a factor from T cells, that it would cause the B cells to switch from IgM synthesis to IgG synthesis, and in particular gamma-1. And so that was called a gamma-1 switch factor. So that was 82. Now, it turns out that Toshio Hirano, who is the first author of this paper that we're going to review today, was a young postdoc, or a postdoc in 1977, working with Professor Yamamura at Osaka University. And at the same time, Kishimoto was there. He'd gone back there. And Hirano, rather than work on, and I guess Yamamura, rather than work on mouse B cells, they decided they wanted to work on human B cells. So they worked up a system where they activated human B cells with pokeweed mitogen. Pokeweed mitogen, I haven't talked a lot about that in this journal club, but it was like PHA for T cells. Pokeweed mitogen would activate T cells and B cells and cause the B cells to increase the production of immunoglobulin. Hirano was introduced then to this whole T cell replacing factor, or T cell factors that worked on B cells. In the introduction to this paper, they mentioned earlier, right off the bat, that they had started purification, or that Hirano had started purification after he left Yamamura's lab in Osaka. He went to a hospital there in Osaka, and he set up his lab, and he started trying to purify this factor in 1978. That's key in terms of who's responsible for what in this whole process. And he worked on that for 6 years in this hospital in Osaka. And then he joined Kishimoto. By this time, Kishimoto had become somebody in the business, had his own lab at Osaka, and he was looking for somebody like Hirano, who was really a good biochemist, to purify the factor that he had been looking at. And so Hirano got a promotion and joined Kishimoto's lab as an associate professor in 1984. Very rapidly thereafter, they discovered or created a human T cell line transformed by human T leukemia virus, HTLV1, so that it would grow autonomously. It didn't need IL-2 or anything like that. And this cell line made 900 times more of their factor than they could get from normal human T cells. So that was key, because then what Hirano went on to do was that he went on to purify this factor to homogeneity, showed that it was homogeneous. There weren't any other protein molecules left, separated them all away with a series of biochemical steps. And he got to the point where he could then determine the amino terminal sequence to show that he only had one amino terminus in his preparations. So he published a paper in 1985 to that effect. And so they decided what they needed to do was that they needed to see if they could identify a cDNA clone that would code for the protein of this factor. And so that's what they set out to do in this 1986 Nature paper. So they purified what was then a huge amount of this in the materials and methods now we are in. And they got 1 nanomole, and it doesn't sound like very much, 1 nanomole, but that was 20 micrograms. And that was how much we needed for 1 immunization of 1 mouse to generate a monoclonal antibody reactive to interleukin-2. We figured we needed about 15 to 20 micrograms. So they took that and they tried to clone the cDNA from a cDNA probe specified by the amino terminal 10 or 15 amino acids that they had found that they'd already sequenced from them. And they worked and it didn't work. They couldn't convince themselves that they could find the cDNA. So they figured they needed a better experimental system. And so what they did was that they digested their purified protein with lysyl endopeptidase. So they would clip the things into different peptides. They would then purify the peptides and sequence each peptide. And then they purified them with HPLC, and then they sequenced them. And on the basis of the amino acid sequence, they could then see what predicted DNA would be for that. And they could make oligonucleotide probes, radiolabel them, and then they could probe a library that they also created through the messenger RNA extracted from their cell line. So that's what they did. In order to their assay for what they call BCSF2, B cell stimulating factor 2, they assayed them on a human B cell line, Epstein-Barr virus transformed, and they would add their factor culture for 3 days, and then they would take the supernatants from those cells and measure the IgM concentration with an ELISA. And in order to get protein to see whether or not it had biological activity, they would transfect their cDNAs into COS7 cells and then wait for a while. And so the COS7 cells would transcribe and then translate and secrete immunoglobulin molecules that they could then assay in their BCDF assay, BSF2 assay. So that brings us to the results and to the screen sharing. And so here on the left is figure 1. And in the A portion of this figure are 7 different peptides that they had sequenced. And this is the amino acids, symbols for the amino acid sequence of all these peptides. And then in B, they could use the sequences to then predict a cDNA probes. And so they had from the N terminus, from 6 to 11 amino acids here, they had 3 probes, A1, A2, and A3. And then this is the third, they had 9 different fragments. And here they made B1 through 6. And then fragment 8, and they had C1 and C2. Now they from the N terminus, they were able to find that they were ultimately able to find, probing with these different probes, they could find a cDNA that they thought was their money man, and it was just labeled number 38. It hybridized to the A2 probe, the B2 probe, and the C1 probe, but it didn't hybridize to anybody else, or the probes didn't. None of the other probes worked. So that they then took that 38, the cDNA, and they rescreened the libraries again, and they got 5 clones greater than 1 kilobase long. And that's what they then went to and characterized further. So then in the figure 2, we have 3 of the positive cDNA clones that had then been transcribed and translated into cause cells, and the cause cell supernatants then measured in the reciprocal dilution of the soups into the concentration of IgM through the ELISA. And these were 3 positives, these are 3 negatives. They also raised a rabbit antibody to the N-terminus, and they constructed affinity columns, and they could show that their rabbit antibody would take out the activity that they were measuring. So then they did, they looked at the sequence of these cDNA clone, and they had in B here of this figure, the amino acids are on the top, the nucleotides are on the bottom, and the hold on, I got to get to my notes here. From this, the amino acid sequence data and the N-terminus, they figured they had an open reading frame of 184 amino acids that would give a predicted size of 20.78 kilodaltons. And Toshio had already shown that in his purified protein that on gel filtration and SDS page gels, it was 21 kilodaltons, not bad for a biochemist. And then they went on further and they looked in the databases to see whether or not they had any homologies with any other cytokines that had been structurally determined. And they found they found one, it was GCSF. And so that was good too. And then they took their cDNA probe, ready labeled it, and they did northern blots. And this is for on unstimulated T cells. This is B is PHA stimulated normal T cells. C is their T cell line that they had used to derive all of the data and the cDNA. And it's interesting, D and E, this is a bladder carcinoma cell line that they had found that made a lot of this factor. And then this is a cardiac myxoma cell line where patients with cardiac, this is a benign tumor from the heart was making a lot of this factor. And the interesting thing is that these people had a lot of autoimmune phenomenon. So that's it. That was it. Of the data in this paper, and so subsequently, all kinds of things happened, and I wanted to draw your attention to this particular publication because it was edited by me. This is called A Living History of Immunology, and there's more than 20 different individuals that when I was the chief editor-in-chief of Frontiers in Immunology, I decided to ask all the immunologists who were still living and well to write, just a few pages on how they got to be famous and how they became successful. And Toshio has a great paper in his Frontiers in Immunology, and so I recommend it to you highly. You should read that. You'll find out, there's always background stories that never make it into the publications. And so you can learn a lot from the publications, like we're trying to do today and in this journal club. But then you also need to talk to the people. And if you talk to them, you'll find out all kinds of things you didn't know. And in this particular, this is an ebook of the living history. You'll find all kinds of things out. So I'll stop the screen share at that stage of the game. But I want to highlight some of the things that went on. So this molecule ultimately there was a lot of controversy going on in this business, as I've said before. This was November 86 when this paper came out with the sequence of this T cell replacing like factor. It turns out there was another paper that was published in the European Journal of Biochemistry, I think, from a group in Belgium, where they had cloned and sequenced another factor and that had been induced by interleukin-1 stimulated fibroblasts. And that turns out had exactly the same sequence. Sort of strange. T cell derived factor, fibroblast derived factor. And then that was in September of 86, before Toshio's paper. And then in October of 86, there was another one that came out that was thought to be an interferon, but it had the same sequence. And so suddenly there were three different groups that found this thing. And on the basis of all of the different characteristics, the scientists got together and they decided to call this molecule interleukin-6. Then just the next year, there was another investigator, Galdi, who had been working on a factor that was derived from macrophages that stimulated hepatocytes, liver cells, to make all kinds of proteins, which the medicos call the acute phase proteins. One of them is C-reactive protein that reacts to the carbohydrates capsule of pneumococci, and the clinicians use this CRP as an index if they suspect that there is an inflammatory process going on. They look at that and it's usually sky high. Well, interleukin-6 stimulates the liver cells to make this stuff, this C-reactive protein. Also, fibrinogen is controlled by interleukin-6, and a bunch of other what are acute phase proteins that you can measure in the blood and in the plasma of patients. Toshio then had a paper out, I think in 1987, that it could very well be that interleukin-6 was involved in the pathogenesis of rheumatoid arthritis. Independent, subsequently, other papers showed independent of antigen-activated T cells. And so there was something funny going on with autoimmune inflammatory diseases and interleukin-6. And in the plasma of patients that have some sort of a funny inflammatory situation like rheumatoid arthritis, interleukin-6 is one of the easiest cytokines to measure because it's there in the plasma in a very high concentration. Whereas other ones, even like TNF or interleukin-1, they're also there in inflammatory syndromes. And that's what I call the pro-inflammatory triad. The most recent publication that has brought interleukin-6 into the upfront, so people might notice it. If you've been following immuno-oncology and the new immunotherapeutic approaches to cancer, the phenomenon called CAR T cells, CAR, which stands for chimeric antigen receptor T cells. What they do is they take T cells out from a patient and they put in a new T cell receptor in those cells that is programmed that has an immunoglobulin outside part of the T cell receptor and an inside part that's the activating part, and that is from a T cell. And they grow these cells up in large quantities outside the body using interleukin-2. And then they infuse these back into cancer patients, and they've had some remarkable successes and really good for some patients. Not all, but some, some basically ostensibly looks like they're cured. Like everything else in life, you never get away scot-free. And as a consequence of loading up the patients with billions of these CAR T cells, and then they recognize the tumor cells, and that activates the CAR T cells. And what do? They don't make antibodies, they make cytokines, right? So you get what now the clinicians, every generation of clinicians has their own acronyms and so forth. They're talking about cytokine storm because the patients get really, really sick. Basically, they go into septic shock. And they go into septic shock because the cells, the T cells and the macrophages and everybody is so hopped up from the antigen-antibody reaction that they make the pro-inflammatory cytokines, interleukin-1, TNF, and IL-6 An anti-IL-6 receptor alpha chain antibody in the CAR T cell story has been very effective in dampening down the cytokine storm symptoms, which can kill the patient themselves, essentially. So you might kill the tumor, but you kill the patient at the same time. Now, that's not the end of the story yet, because COVID, as you know, very well know, and what I've been harping on in Twitter and so forth, is that COVID, the symptoms of COVID with the massive inflammatory reaction that occurs in the lungs, which fills up the fluid, the lungs with inflammatory fluid and so forth, that's not caused by the virus. It's caused by the host reaction to the virus, and it's caused by the same pro-inflammatory cytokines, interleukin-1, TNF, and IL-6. And anti-IL-6 receptor has been shown to be effective if given early enough in the whole process. It's just like this new drug out from Pfizer that you got to give it as soon as a patient has symptoms, because they've been generating large amounts of virus for a week to 10 days before they even have symptoms. Same story with anti-IL-6, but that's just one more aspect of this whole interleukin revolution. So I'll stop there, and don't forget to like the video and click on the bell and check out the website and check out the social media. It's been great. Thank you for listening and watching.